using the present.
Okay.
Okay.
Ladies and gentlemen, good afternoon. My name is Guo Xinxin, a PhD candidate from Southeast University in People's Republic of China. It is an honor to participate in this conference and present my research. The title of my presentation today is Fusobacterium, Periodonticum, BCT, and its virulence protein BCT promote esophageal cancer progressing through mast cell MC2 polarization. This work was completed under the supervision of Professor Liu Ran. Esophageal cancer is one of the leading causes of cancer-related deaths worldwide, and understanding its underlying mechanism is crucial for developing effective therapies. Recent studies have highlighted the protection role of the microbial and immune cells in cancer progressing. In particular, FP has been implicated in various cancers, including colorectal cancer and esophageal cancer. In this study, we focus on the relationship between Fusobacterium periodonticum, BCT, and mast cell activation in the progression of esophageal cancer. Mast cell as a group of multipotential innate immune cells originate from bone marrow, differentiate and mature in peripheral tissues, and mainly distribute in digestive tract, reproductive tract, and other parts of the body in contact with the outside world. In the past, mast cell research has mainly focused on allergic reaction, but many studies have shown that mast cells, as a class of innate immune cells, also play an important regulatory role in the microenvironment of different tumors. However, the distribution, immune regulation mechanism, and the specific effect of mast cell in the developing of esophageal remain unclear. So, this study aims to explore the role and the mechanism of FP and BCT virulence protein on mast cell activation and polarization in esophageal progression. First, we detected the presence of BCT in the standard stream of FP. Using RT-qPCR, we examined the expression of BCT in esophageal cancer tissues and adjacent tumor tissues. As figure said, the result showed that the expression of BCT was significant higher in cancer tissues compared to adjacent tumor tissues, especially according to the clinic diagnostic criteria for esophageal cancer. The expression of BCT was higher in patients with more advanced stages. So this result showed that BCT may be related to the development of esophageal cancer. Meanwhile, based on the TSECO database, we analyzed the tumor immune infiltration microenvironment of esophageal cancer and found that activated mast cells were more prevalent in the esophageal cancer patient. We further validated mast cell infiltration in cancer and adjacent tumor tissues from esophageal cancer patients through RT-qPCR and Western blot experiment. As figure 8, the result showed that the expression of mast cell activation marker genes was significantly higher in cancer tissues. As table 2, the mast cell was higher infiltrated in patients with more advanced stages, and Pearson analysis results showed that BCT was positively correlated with mast cells in the esophageal cancer. Next, we conduct cell and animal experiment to investigate the role and mechanism of mast cell in esophageal cancer after the action of FP and BCT. First, we constructed recombinant BCT protein and used BCT as an exogenous substance to treat mast cell. We applied two cell lines, including HMC-1 and primer cells, BMC, derived from multiple mirror cells. The CCK8 assay was used to analyze the effect of FP and BCT on mast cell activity in order to determine the most appropriate concentration for treatment. The result showed that FP increased mast cell activity at more of 100, and BCT increased mast cell activity at a concentration of 100 nanograms per. At the same time, by analyzing the release of beta hexaminidase and using toluidine blue staining to observe mast cell degradation at different concentrations, FP and BCT. The result showed that FP and BCT increased the release of beta hexaminidase and activated mast cell. Next, we established a culture model of mast cell treated with FP and BCT together with esophageal cancer cells, EC109, to investigate the role of mast cell in esophageal cancer. On Figure 15, the transfer assay result demonstrated that mast cell treated with FP and BCT promoted the invasion and migration of esophageal cancer cells. On Figure 16, the colony formation assay showed that mast cell treated with FP and BCT increased the activity of esophageal cancer cells, especially on Figure 70. In the mouse xenograft experiment, it was shown that mast cell treated with FP and BCT promoted tumor formation. This result and demonstrated that FP and BCT activated mast cell to promote the progression of esophageal cancer. To explore the mechanism by which FP and BCT promote esophageal cancer progression, we performed transcriptomic sequence. The result indicated that compared to the control group, 1,312 genes were upregulated and 920 genes were downregulated in the BCT group. Goal function analysis result showed that the PLC gamma PKC pathway was involved in the biological process. Therefore, we used Western blot to verify the expression of PLC gamma PKC pathway in mast cell after treated with FP and BCT. FP and BCT significantly increased the phosphorylation levels of PLC gamma PKC. As Figure 19, the result showed that FP and BCT activated the PLC gamma PKC pathway in mast cells. To investigation the role of the PLC gamma PKC pathway in mast cell mediated esophageal cancer progression, we added the PLC special inhibitor U73122 and analyzed the expression of PLC gamma PKC related proteins through Western blot experiment, as well as its impact on the invasion and migration ability of esophageal cancer cells. EC109. As Figure 20, Western blot identified that U73122 inhibited the PLC gamma PKC pathway of mast cell, which regulated by FP and BCT. As Figure 21, transfer assay identified that U73122 inhibited the invasion and migration of EC109, which promoted by FP and BCT treated mast cell. Based on the above result, we draw the following conclusion. 1. We demonstrate that mast cell and BCT are highly infiltrated and highly experienced in esophageal carcinoma, as well as the positive correlation between mast cell and BCT. 2. Fusobacterium, Periodonticum, and BCT inactivated mast cell. 3. FP and BCT regulated mast cell promote invasion and migration of EC109 by PLC gamma and PKC pathway. That concludes my presentation. Thank you to all the team members and thank you for your listening.